Welcome to another fun episode by the Dog Mind Gears podcast.
Glad to be here for this one.
Yeah, so I want you to put yourself in a really difficult position for a second.
Okay.
Just imagine you're sitting in a sterile veterinary clinic. You hear the hum of the fluorescent lights buzzing above you.
Right, that awful sound.
Exactly. And you've just been handed a terminal diagnosis for your best friend, your dog. What would you do?
I mean, most of us would be completely devastated.
We'd probably ask about palliative care, just to keep them comfortable.
Start preparing to say goodbye.
Right. But today, we are looking at an incredible story from Dogazilla magazine about a tech entrepreneur who got that exact death sentence for his dog, and, well, he didn't accept it.
Not at all.
Yeah.
No, instead, he used ChatGPT to literally hack a cure.
Which sounds like science fiction, but the mission for our deep dive today is to really break down the mechanics of this. We're going to explore how a biological tragedy was basically reframed as a data problem.
Yeah, a pure data problem.
Exactly. We are going to look at the exact pipeline that took raw tumor data, processed it through artificial intelligence, and resulted in a $3,000 custom RNA vaccine.
Which is wild.
It is. And more importantly, we need to examine what this specific pilot study signals for the future of personalized medicine.
For pets and humans, right?
Yeah, not just for the pets sharing our homes, but for human medicine down the line as well.
Okay, let's unpack this. Because before we even get into the weeds of artificial intelligence and RNA synthesis and all that, we really need to understand the massive paradigm shift that made this whole thing possible.
Right, the fundamental change in perspective.
Yeah. So this happened in late 2025. Paul Conyngham, who is a tech guy living in Sydney, gets the absolute worst news. His 8-year-old dog, Rose, has this aggressive terminal cancer.
And she's given months to live.
Months. Just the ultimate gut punch,
Oh, absolutely. And it really highlights a very specific wall that traditional veterinary medicine often hits.
Yeah, the limits of standard care.
Exactly, because the standard protocols like chemotherapy and radiation, they are powerful tools, obviously, but they are built on a statistical model.
Right.
They're generalized weapons designed for the average patient based on decades of broad data.
So they aren't personalized at all.
No, not at all. When you administer those treatments, you are basically flooding the body with systemic toxicity.
Hoping the cancer dies just a little bit faster than the healthy cells do.
Right, it's a race. And for an older dog like Rose, well, her body simply couldn't handle that level of collateral damage.
Because traditional oncology in this context is essentially treating the cancer as a uniform biological enemy.
You're throwing a really wide net.
Exactly.
Yeah.
But Conyngham looks at this, and because of his tech background, he makes a radical pivot.
He does.
He decides he isn't going to look at Rose's tumor as this sad, inevitably fatal biological specimen. He decides to look at it as a digital sequence.
And that right there, changing the battleground from the biological realm to the digital realm, that is what dictates the speed of this entire process.
Because data moves faster than biology.
Precisely. Conyngham utilized rapid DNA sequencing. So instead of just looking at the tissue under a microscope to determine the type of cancer...
Which is the normal route.
Right. He took a piece of that physical tumor and ran it through a sequencer.
Okay, so what does that actually do?
This machine essentially scans the physical matter and outputs gigabytes of raw text files.
Wow. Literally turning flesh into text.
Yes. We are talking about converting a chaotic physical malignancy into a highly readable, organized digital blueprint.
Containing, what, billions of base pairs of genetic data?
Exactly. Billions. And once a tumor exists as a digital data set, you can apply massive computational power to it.
So it's kind of like you're dealing with a locked safe, right?
I like that analogy.
The traditional approach, the chemotherapy approach, is basically trying to blow the safe up with dynamite.
Right, blunt force.
Yeah, you might get it open, but you're going to destroy the room in the process, which in this case is the dog's body.
Unfortunately, yes.
But by converting the tumor to data, it's like you're sliding a microscopic high-tech scanner inside the safe to map the internal mechanics.
To find the exact combination.
Exactly. And then you unlock it without breaking a single thing.
That's the ideal scenario, yes.
But here's the thing that really trips me up. You scan the safe, right? You get the blueprint.
Right.
It's just a massive data set of genetic information. How does a tech guy read a biological blueprint of that magnitude?
Well, the short answer is he doesn't.
Oh.
When dealing with genomic data sets, manual analysis is computationally impossible for a human brain.
Because of the size.
Yeah, a single tumor genome contains billions of letters of genetic code, and the mutations driving the cancer are often just a handful of misspellings hidden within that massive library.
Like finding a needle in a digital haystack.
Worse than a haystack, honestly.
Yeah.
But we have to view DNA fundamentally as a language.
Okay, a language like English or Spanish.
Sort of. It has syntax, it has grammar, and it has structural rules that dictate how a biological organism operates. You've got the letters A, C, G, and T.
The basic building blocks.
Exactly. When cancer occurs, it is a grammatical error in that code telling a cell to multiply uncontrollably.
A glitch in the software.
Right. So, finding that error requires a tool designed to parse massive amounts of language-based data at lightning speed.
And this is where the story takes a wild turn.
It really does.
Because to find that typo, Conyngham turns to ChatGPT.
Yep, off-the-shelf AI.
He takes this raw genetic data set, plugs it into the AI, and uses it as a high-speed linguistic debugger for his dog's DNA.
Which is brilliant when you think about it.
It's insane. He's asking it to scan the genomic landscape and pinpoint the unique mutations, the specific glitches that are driving Rose's individual cancer.
And large language models like ChatGPT are, at their very foundation, advanced pattern recognition engines.
Right. They predict the next word.
Yeah, they're trained to predict and identify linguistic patterns based on context. While we normally use them for English or, Python code, genetic sequencing is just another syntax.
It's just letters in a row.
Exactly. So when you feed it tumor data alongside healthy baseline data, the AI applies its pattern matching capabilities to filter out the noise.
It compares the two files, basically.
Right. It isolates the anomalies, the deviations from the healthy grammar, if you will, and it flags those specific genetic mutations as the drivers of the malignancy.
Okay, wait. I have to play the skeptic here for a second.
Go for it.
ChatGPT is trained on internet text, right? It aggregates Reddit threads, Wikipedia, books.
Yes, massive amounts of web text.
It doesn't have a medical degree, and it certainly isn't a veterinary oncologist.
True.
How does it know how to process the syntax of genetic sequencing without just hallucinating or making things up entirely? I mean, we've all seen AI confidently give completely wrong, even unhinged answers.
This raises an important question, and it's the exact reason why this project couldn't just happen in a guy's garage.
Right, because of the risk.
Exactly. If an AI hallucinates a genetic target, meaning it flags a healthy, normal genetic sequence as a cancer mutation, the results are catastrophic.
What happens then?
Well, if you build a vaccine based on that false target, you will train the dog's immune system to attack its own healthy tissue.
Oh, wow.
Yeah, you would induce a severe, potentially lethal autoimmune disease.
That's terrifying.
It is. The AI is brilliant at filtering the data set and spotting anomalies quickly, but that digital insight is merely a hypothesis.
So it's not the final answer.
Not at all. It must be subjected to rigorous human scientific validation.
Okay, so the AI prompt isn't the cure itself. It's just a heavily educated guess that needs to be fact-checked by real scientists.
Precisely. And the next phase of the story, the academic collaboration, is where those safety mechanisms are actually applied.
Right, because he didn't do this alone.
No, Conyngham partnered with Professor Paul Conyngham and the UNSW RNA Institute over in Australia.
Which is a major research hub.
Huge. And the scientists at the university took the mutations that ChatGPT flagged and ran them against known, validated biological databases.
Like checking the AI's homework.
Exactly like that. They used things like the Canine Genome Project. They had to manually ensure that the genetic targets the AI found were actually oncogenes.
Meaning known cancer drivers.
Right. And that they were biologically actionable. They acted as this vital institutional safety net, translating the AI's rapid data synthesis into scientifically sound medical targets.
That dynamic is just incredible to me. It's the ultimate team-up between a citizen scientist and an academic institution.
It's a very modern partnership.
Yeah, the tech entrepreneur brings this insane speed and a high risk tolerance, and the university brings the rigor, the safety protocols, and the literal laboratory infrastructure to error check the AI.
Because without that error checking, you have nothing safe to use.
Right. And once they validated the data and they confirmed the AI didn't hallucinate, they had to actually build the thing.
The physical vaccine.
Yeah. Can you break down that pipeline? Like, how do you physically go from a verified digital text file to a vial of medicine?
So the pipeline relies on synthetic messenger RNA or mRNA.
Which we're somewhat familiar with now.
Yes, thanks to recent years. Once the exact genetic sequence of the tumor's mutation is confirmed on a computer screen, the lab uses specialized equipment in a process called in vitro transcription.
So it's complicated.
It basically synthesizes a physical mRNA strand that perfectly mirrors that cancer mutation.
Okay, so they print the instructions.
Right. But you can't just inject raw mRNA into a dog's arm.
Why not?
Well, enzymes in the bloodstream would shred it instantly. It's very fragile. It has to be packaged.
Okay. How do they package it?
The lab encapsulates this delicate mRNA inside lipid nanoparticles.
Lipid nanoparticles.
Yeah, which are essentially microscopic fat bubbles.
Oh, fat bubbles. That makes sense.
These bubbles protect the mRNA courier as it travels through the bloodstream, allowing it to slip safely inside the dog's immune cells.
And once it's inside,
Once inside, the mRNA delivers the instructions, essentially handing the body's natural defenses a highly detailed, wanted poster of the cancer's unique genetic signature.
a wanted poster. I love that. And the most mind-blowing part of this whole pipeline is the cost and the speed.
It really is unprecedented.
Right. Because we are so conditioned to expect medical breakthroughs to take a decade of trials and billions of dollars in funding.
That's the traditional model.
Yeah. And Professor Thordarson himself admitted he was incredibly skeptical when Conyngham first approached him.
Of course he was. He's a traditional academic.
Right, he didn't think an academic lab could move fast enough to save Rose before the cancer took her.
But they did.
They did. From the initial sequencing through the AI debugging, all the way to a manufactured, sterile, lipid-encapsulated RNA vaccine.
The whole thing.
It took less than 2 months.
Unbelievable.
And the out-of-pocket cost was approximately $3,000.
And that speed fundamentally disrupts the current medical paradigm. By treating the cancer as an information problem first, they completely bypass the years required for broad, generalized clinical trials.
They skipped the line entirely.
Yeah, they went straight from data analysis to a custom manufactured biological solution tailored for a sample size of exactly 1.
Sample size of 1. That's the core of personalized medicine right there.
Absolutely.
Okay, so they have the physical vaccine, they administer it to Rose. But immunotherapy isn't like movie magic where the patient gets a shot and instantly leaps out of bed, right?
No, not at all.
There is a real-world biological and emotional timeline here that requires a massive amount of patience.
Yeah, the waiting game is notoriously difficult with immunotherapy.
Because it's not instantaneous.
Right. Unlike a painkiller that binds to a receptor and offers immediate relief, an RNA vaccine requires a biological lag time.
A lag time. How long are we talking?
Well, when the lipid nanoparticles deliver that blueprint into the immune cells, you aren't directly killing the cancer right then and there.
Oh, right. You're just delivering the wanted poster.
Exactly. You are running a boot camp for the immune system. The specialized immune cells, specifically dendritic cells, have to translate the mRNA.
Okay.
Then they display the cancer antigen on their surface, and they travel to the lymph nodes to train the T cells.
That sounds like a lot of steps.
It is. Those T cells then have to multiply into an army large enough to actually mount a systemic defense.
And that takes time.
Yeah, that cellular machinery takes weeks to fully activate.
And you can really see that agonizing lag in Rose's timeline.
It must have been terrifying for the owner.
Totally. In early December, right around the time of the diagnosis, Conyngham describes her as immobile, sad, and completely shut down.
Just a sick, sick dog.
Yeah. They administered the vaccine, and for weeks, it probably feels like absolutely nothing is happening on the outside.
But beneath the surface, her immune system is going through that exact boot camp you described.
Right. And then we hit late January, so about a month or so later, and the physical recovery changes are just staggering.
It's a night and day difference.
Rose goes from being completely immobile to literally jumping over fences to chase rabbits in the yard.
Incredible.
Her energy returns entirely. And when they finally ran the scans, statistically, they achieved a 75% reduction in the cancer.
And we really need to pause on that number. In the context of a terminal, aggressive oncology diagnosis, reducing a tumor mass by 3/4 is an absolute clinical triumph.
It's massive.
It effectively shifts the patient's status from immediate end of life to managed chronic care.
Buying them so much time.
Yes. It stabilizes the biological environment, relieves the pressure on surrounding organs, and removes the immediate lethal threat.
But here's where it gets really interesting, though. And I have to push back a little on this being a total victory.
Okay, let's hear it.
Why only 75%?
Ah, yes.
If the AI successfully found the glitch,
Yeah.
right, and the university synthesized the vaccine to train the immune system to attack that specific glitch.
Yeah.
What happened to the other 25% of the tumor?
It's a great question.
Why didn't the T-cell army wipe the whole thing out?
Because tumor heterogeneity is one of the most complex challenges in oncology today.
Heterogeneity, meaning it's not all the same.
Exactly. We often mistakenly assume a tumor is just a uniform mass of identical cloned cells.
Like a solid block of the exact same bad code.
Right, but it's not. As a tumor grows and divides, it continues to mutate internally,
Oh,
creating these distinct subclonal populations.
so it's mutating while it's growing.
Yes. That remaining 25% represents a subpopulation of cancer cells within Rose's tumor that possessed a slightly different genetic profile than the dominant mass.
Huh, I see.
The vaccine was perfectly engineered to wipe out the main strain of the cancer, but those minor subfactions evaded detection.
Because their specific genetic disguise wasn't on the wanted poster the RNA delivered.
That analogy captures the mechanics perfectly.
So the tumor is almost like a massive criminal syndicate with different factions.
Yes, exactly.
You gave the immune system the profile for the main boss and their crew, and they took them out. But a splinter group with a completely different disguise survived the raid.
That's exactly what happened, and this limitation shows exactly where the future of this technology is heading.
Oh, where is it heading?
The next evolution is multivalent, or combination vaccines.
Meaning more than one target.
Yes. Instead of the AI just looking for the one dominant mutation, the computational power would be used to identify 3, 4, or 5 different genetic signatures representing all the various subpopulations within the tumor.
Rounding up all the bosses.
Exactly. The lab would then print a single RNA vaccine containing multiple different blueprints.
Wow.
Simultaneously cutting off every evolutionary escape route the cancer might try to take.
That is just brilliant. So what does this all mean for us? Because as much as we love a heartwarming story about a guy using tech to save his best friend,
Which is a great story.
it has massive implications. Rose's success is essentially a pilot study. It's a critical proof of concept for something called the One Health Blueprint.
Right. The One Health approach is a vital framework in modern medical research. It recognizes that human health, animal health, and environmental health are inextricably linked.
We're all in the same ecosystem.
Exactly. Dogs serve as the perfect biological sentinels for human cancer.
Sentinels, because they live with us.
Yeah, they live in our exact environments, they drink our water, they are exposed to the same household chemicals, and most importantly, their immune systems develop remarkably similar spontaneous cancers to humans.
The spontaneous cancers, meaning not genetically engineered in a lab.
Right, and they respond to treatments in very similar ways to humans, both genetically and biologically.
Wait, I'm stuck on something here.
Sure.
I get how a dog's tumor is genetically similar to a human's, and the environmental factors overlap. But how does curing a dog actually fast-track human trials in any legal or scientific way?
It's about the regulatory pathway.
Because aren't the FDA regulations and human clinical trial pathway completely separate from veterinary medicine? A success in a vet clinic doesn't magically approve a drug for a human hospital.
You are absolutely correct that the regulatory bodies are separate, but you have to look at the mechanism of comparative oncology.
Okay, break that down for me.
When researchers apply to the FDA for an investigational new drug application, an IND, to begin human trials, they need massive amounts of preclinical safety and efficacy data.
They need proof it won't just kill the patient.
Exactly. Now, traditionally, this data comes from engineered mice.
Lab mice.
Right, which are honestly terrible proxies for complex human immune responses.
Because they're mice.
Yes, but by proving that this specific RNA manufacturing pipeline and the LNP delivery system works safely and effectively in dogs.
Dogs who have complex, natural immune systems.
Exactly, dogs battling real, spontaneous tumors. When you prove it there, researchers generate incredibly high-weight preclinical data.
Oh, I see.
The FDA doesn't accept the dog cure as a human cure, but the overwhelming proof of the mechanism's safety in dogs dramatically accelerates the regulatory approval process to begin human trials.
That makes total sense. The mechanism itself is what is being proven.
Precisely.
And it doesn't even stop at cancer, does it? Yeah. Professor Thordarson speculated that this exact same RNA platform could be adapted for completely different conditions.
It can. The manufacturing pipeline for RNA is fundamentally disease agnostic.
Disease agnostic, meaning it doesn't care what it's treating.
Right. The machinery printing the mRNA strands doesn't care what code it is assembling. Once you have the established laboratory infrastructure to safely print and package custom RNA, You simply swap out the digital payload.
Like changing a software cartridge.
Exactly. Today, we are talking about a cancer blueprint. Tomorrow, that exact same platform could be utilized to deliver genetic instructions to correct anomalies driving complex neurological diseases.
Like Alzheimer's or Parkinson's.
Potentially, yes. Or even to modulate the immune system for severe autoimmune disorders. The physical delivery system remains identical, only the software being loaded into it changes.
That is wild. It really highlights the rise of the citizen scientist in all of this.
It absolutely does.
Because we are seeing highly motivated individuals with tech backgrounds looking at these historically closed-off medical systems and basically forcing a faster, more transparent evolution of healthcare.
They're pushing the boundary.
They are demanding that traditional medical frameworks adapt to the speed of the digital age.
But the democratization of medical research, while it is accelerating discovery, it is crucial to temper that excitement with a clear understanding of the risks.
We can't just have everyone playing doctor.
Right. The collaboration between motivated families and local research labs can be incredibly productive, but it requires strict adherence to safety protocols.
Which is exactly where we should land this. Because if you are listening to this right now and you have a pet facing a terrifying health crisis,
Which is an awful place to be.
it's so easy to want to jump online, fire up ChatGPT, and try to replicate this tomorrow.
Too easy, really.
Yeah, but we need to look at the practical steps and safety protocols based on the realities of this specific source material.
So the first and most critical step is acknowledging that what happened with Rose is currently a one-off, highly experimental treatment.
It's not the norm.
No. Custom synthesized RNA vaccines are not something you can just order off a menu at your local veterinary clinic. It is entirely outside the realm of standard care.
Right. Step 2 is you must start by consulting your licensed veterinarian.
Always.
You need to ask them specific questions about the limits, the potential benefits, and the absolute reality of traditional care for your specific pet's condition before you look anywhere else.
You have to know what the standard baseline is before you deviate from it.
Exactly. And step 3, if you do decide to explore experimental research paths, you cannot do it alone.
Never alone.
As we discussed, the AI is a flawed tool.
Very flawed. You must coordinate effectively with local research universities and academic institutions.
You need that safety net.
Yes. The clinical oversight, the biological error checking, and the sterile manufacturing provided by a university lab are non-negotiable requirements for safely turning a digital idea into a biological reality.
It's all about balancing that tech-driven hope with rigorous scientific safety.
Beautifully said. And if we look toward the horizon, there is a final thought I want to leave you with today.
Let's hear it.
If a tech guy using an off-the-shelf AI tool and just $3,000 can successfully hack a custom genetic cancer vaccine for his dog, what happens to the massive multi-billion-dollar global pharmaceutical industry when the tools to create highly personalized individual cures are eventually sitting on everyone's desktop?
For more information, visit the original source article by Dogazilla magazine at dogazilla.com.